Hello, everyone. Welcome. Thank you so much for joining us today. We'll let everyone gather and just get started in a few seconds. Thank you for everyone for coming today. Welcome. All right, again, thank you everyone for joining us today. Welcome to the Monarch Conservation webinar series. Today's webinar is Habitat Monitoring with the Monarch Joint Venture. Our presenters today are Alex Palmerlee with Farview Ranch and Red Boot Ecology, and Jennifer Thieme and Alex Carroll from the Monarch Joint Venture. My name is Jenna Tots. I'm the MJV Education Coordinator, and I'm joined by my coworker, Alyssa Taylor, Program Coordinator. We want to thank our sponsor for their support of this webinar series. Our supporting sponsor is Reinvent Your Waste. We so appreciate your support and dedication to monarch conservation. If you or your company is interested in sponsoring this year or next, please email learn@monarchjointventure.org. Before we get started, we have a few community guidelines and helpful hints for this webinar. If you're joining us today but didn't pre-register for the webinar, we'd like to count you for attendance and be able to send you some follow-up resources. Again, you can email learn@monarchjointventure.org with your name, city, and state. This webinar will be recorded and posted to the Monarch Joint Venture website by Friday of this week, but usually I get it up by Wednesday or Thursday. The entire Monarch Conservation webinar archive is free and available to watch. There is over 10 years of amazing content that you can watch at any time. During the webinar, please put your questions in the Q&A box. You can find that by going to the bottom of your Zoom screen and seeing Q&A. If you don't see that, there should be 3 dots. It says more. Please click on that, and you should see a Q&A box. Please don't use the chat. It's just easier for us to collect all of those questions, for the end for a Q&A session. Please use reactions also during the presentation. Again, these are found at the very bottom of your Zoom screen. If you can't find them, again, click on that 3 dots to see more. Yes, I see people are practicing hearts and thumbs up. This is a great way to be able to interact in some way with the speaker. I know that the speakers can't see you, so this is a fun way to do that. We do want to remind you that there are translated captions for this Zoom call. If you'd like to add captions in a language other than English, you can click on the arrow on captions, again, at the bottom of your screen. Be sure the translation button is on, and then you can choose your caption language. And finally, all of our webinar speakers represent their own viewpoints, which are not necessarily those of the Monarch Joint Venture. And now I will introduce our speakers. I'm going to introduce all 3 of them, and then we will toss it back over to Alex to get us started. So through his work at Farview Ranch, Audubon, California, and the Center for Land-Based Learning, Alex Palmerlee has over 20 years experience implementing restoration projects in California's Central Valley and surrounding rangelands. He received an MS in Ecology from UC Davis. His research has focused on cost effectiveness of restoration practices, interactions of cattle and blue oak woodlands, and best practices for implementing pollinator plantings on cattle ranches. Jennifer cultivates the MJV's network of researchers, research entities, and partner science initiatives to collaboratively advance monarch conservation research. She also coordinates MJV's research and monitoring projects throughout the U.S. She works with the MJV science team to maintain the Integrated Monarch Monitoring Program, or IMMP, encourage its use, and share findings from the program with the broader conservation community. Jennifer received her BS in Environmental Science from Carroll University in Wisconsin, and her MS in Fisheries and Wildlife Biology from The Ohio State University. Prior to joining MJV in 2018, Jennifer worked on a variety of grassland bird and vegetation research projects and for several conservation organizations working to protect water quality and wildlife habitat in the Midwest. In her spare time, she enjoys birding, enhancing her urban prairie garden, and challenging herself with literature in Spanish. And as Eastern Science Coordinator, Alex Carroll advances monarch conservation by supporting MJV's science and monitoring initiatives. Through the IMMP and remote sensing program, she conducts pollinator and habitat surveys to track monarch populations and evaluate the availability of milkweed and nectar resources on the landscape. Alex is from St. Paul, Minnesota, but she fell in love with prairie ecosystems during college at the University of Minnesota Morris, where she graduated with a BA in biology and environmental science. Before joining MJV, she conducted habitat restoration with native resource preservation, and she researched the effects of prescribed burns on prairie plants with the Ecology project. In her free time, Alex can be found baking, playing board games, reading fantasy novels, climbing trees, and searching for insects. We are so happy that you are all here. I'm going to stop sharing my screen and I will pass it over to Alex.
Thank you, Jenna.
Of course.
All right.
Looks great.
So my family owns and manages Farview Ranch, which is a 1,000-acre cattle ranch in the foothills of Northern California. And we worked with Monarch Joint Venture, California Resource Conservation Districts, California Association of Resource Conservation Districts, National Fish and Wildlife, and Bureau of Land Management to do a series of pollinator implementation projects at the ranch and also on a few other sites. And we're just going to report on sort of the process of those projects, how we went about those, and some best practices and hard-earned lessons that we had along the way. So we did, this is sort of two projects. One is what we called the Pollinator Islands. We have on Farview Ranch, we have a number of our 1,000 acres was intensely managed for agriculture in the late early 1900s. And many common species that are common on other cattle ranches that neighbor us are not present on our property. So one of the main goals for this project was to get some of those more common pollinator plants back on the landscape. And so we did that by over 3 years. We planted 60 individual plots measuring 10 feet by 100 feet. The idea for this was really to try to cover as much space of that 1,000 acres without obviously restoring the entire 1,000 acres, which, while possible, would break the budget. Each year we collected 6 pounds of seed from our neighboring ranches that have these common species by hand. So that was a big endeavor, but it was important to us to get some hyper-local ecotypes from local plants. The second project that I'll be talking about was with Asclepias cordifolia, heartleaf or purple leaf, purple milkweed, which is a very strange and charismatic milkweed. It's very sporadic across the landscape. It's in small colonies with maybe only a few plants, sometimes 20 to 50 plants, but those are certainly the exception. It grows in hillsides, on flats, and it's a very early emergent milkweed. So for us in California, that became a really key aspect of that plant and has made it a species of interest. However, it is not available really at all that I know of through commercial nurseries. There are some people that helped me plan and think about this project that are probably on this webinar and know much more about this species than I do. So they can chime in the Q&A. So here we are, California up there. We're about an hour north of the capital, Sacramento. This is a picture of the ranch there. You can see the smattering of plantings that were really meant to act as sort of islands of infused plant species that could then on their own sort of seed out as they need to, as the cattle get them stuck to their skin and hair and trample around the rest of the ranch. And we wanted to see if we could do this without keeping cattle out of areas. So this area in the middle of the screen here, this triangle, you can see kind of 2 creek systems. That area had been fenced off from cattle about 20 years ago as part of other restoration work that we'd done on the ranch. So we were able to do half of these plantings every year, half with cattle, half without cattle, sort of just be able to compare if it's even possible to do pollinator restoration with cattle. The reason for that is that the reason we even knew that our plants, that our ranch was void of these plants, was that these plants are coexisting with cattle grazing on our neighboring ranches. So we knew that a specific suite of those species were definitely copacetic with grazing, which, you know, we're interested in obviously restoring the biodiversity of our ranch, but also on a larger landscape level, wanting to offer practices that are practical for other landowners, and fencing off large areas from cattle. If you can avoid that step, then it's a benefit. So here's our seed mix, which will probably only mean things to people that are from California. You'll notice that there's 3 native milkweeds that are in the mix. 2 of those are on the property already. So the purple milkweed, which I spoke about, we're lucky enough to have a few colonies of that. Narrow-leaf milkweed is certainly by some considered a weed. It's very hardy. And then the Indian milkweed, Asclepias areocarpa, is common in colonies on neighboring ranches, but is a species that we have since brought into our ranch. But as of 5 years ago, there were none of it on our ranch. All right, so each year in the fall, you know, California gets this very Mediterranean climate. It's a very bipolar existence. We get a rainy season between October, November, going until shorter and shorter every year now. Used to be through May, now kind of through April, mid-May. And then we get 0 rain, no rain at all, not an inch, not a drop between June and when the rains come back in the fall. So after those first germinating rains, which means that the annual grasses that were brought over by European settlers that have sort of taken over the grazing landscape from the California perennial grasses, all of those annual grasses come up in a big flush. And we can kill that first flush of weeds with a disc. And so we wait for that first germinating rain, and then we cut down all that seed bank. One of the challenges with this area, it's in the foothills out of the Valley of California, so they're much drier conditions. And that means the benefit of that is that once we've killed that first round of weeds, there's not a lot of germination of more weeds, so that in some areas that's a sufficient amount of weed control. The unfortunate part of that is that it's pretty tough conditions to get seedlings established. So like I said, we collected all the seed by hand, and then we weighed out all the individual species. So I would, you know, weigh out one species and put it into 20 different bags, weigh out another species, put it into 20 different bags. So at the end, we had 20 bags of this of equally weighed seed mix, right? Because we wanted to be able to say that we had put the same amount of seed in the same amount of area in the grazing versus ungrazed areas. Then we used the chain harrow, which you can see on the right, just attached to an ATV. Harrowed that over. It basically just kicks apart some of the dirt clods, and you know, the seed is pretty small. It doesn't need to be buried really at all. Some of the seed is so small that you could just press it into the soil. And then we did nothing after that. So the rain came, germinated things, and we just stepped away, which is very atypical for a lot of restoration projects. If you're doing a hedgerow, you know, it's in a strip, you've got a row of irrigation. Again, this is 60 plots over 700 acres of our ranch. So doing a lot of post-planting interventions just wasn't practical. So for the monitoring, every year we would go out in the spring and just see if things were germinating, just confirm that the seeds had sort of passed that initial test. And then we would do another monitoring toward the end of the summer to find out what was flowering, what looked like it had perennialized. And so we did that this was just our 4th year of monitoring for this project. So the results are here on the left, pretty bad results. Definitely not what we were expecting. This gum plant species is a very common species. While it was not on our ranch when this project started, it's very common across the landscape on other ranches. Media is similar, and buckwheat we've had on the ranch, but not widespread. So again, I'll just about the monitoring protocol. We basically ran one line down the center of the planting area, walked 10 steps, and put down this PVC square and counted individuals that were in that square. So it's possible that there were other species that were out of that monitoring area, but just from from the eye test, I would say that this is a pretty fair representation of what's out there. So the reasons behind this, we had some crazy years in this 3-year stretch, which certainly from a climactic standpoint is becoming more of the norm. The first year we had a really dramatic midwinter drought. So while we have a rainy season during the winter, it's pretty common and is historically common that we have a week or 2-week period where you get no rain, sometimes 3 weeks. In the last 10 years, we've had 5-week, 6-week, 8-week midwinter droughts. And that no doubt has a huge impact. This is usually happening in January. We're implementing in November, those first rains, November, December. So these species will germinate in 2 weeks. In 2 weeks, these guys are up. They're germinated, they're making their go for it. So you imagine they've got their roots down just a couple inches into January and the faucet turns off for 5 to 8 weeks. There is no moisture in the top few inches of soil. So that had a really big impact. Year 2, we had great rainfall, really, really good, which is why we wanted to do this over multiple years, because we know this about this area. It has big swings. But it was followed by a bad grasshopper year, which, we did the spring monitoring. Things looked awesome. They were going to write songs about us. And then come the summer monitoring, most of it had been eaten by grasshoppers. But as it turned out, in some of the subsequent monitoring, year 2 was not more of a disaster than year 1, because the grasshoppers hit late and most of the seeds had either been able to set seed or to perennialize. Year 3, however, we had an insane grasshopper outbreak. I've only seen that one other time in my life. They ate basically anything that was green, weeds that you can't pull without leather gloves. They ate these weeds, stems, leaves, and all. So it's hard to exaggerate how extreme this was. If some of you are in the Midwest, I've heard Mormon crickets used as an analogy to what happened to us last summer. Okay, so I'll talk more about the key takeaways at the end. I'm going to switch gears and talk about this Asclepias cordifolia ASCO trial that we did both on a BLM property and on Farview Ranch. As I mentioned at the beginning, this milkweed in particular became of special interest because of its early emergence and playing a key role in that early monarch migration through northward through California. But aside from seeing it on my property and not even knowing what it was, I remember as a 14-year-old coming across this wild looking plant that I'd never seen before. And of course, I picked the flower, unfortunately, at 14 and took it back home to ask what it was. My dad had no idea. So that's how odd it is to see this plant on the landscape. It's even more odd to see it in a commercial setting. I've been doing restoration for over 20 years and have never seen it on a plant list in any nursery that I've sourced from. Occasionally you can find seed, but again, not common. So it became this question of, okay, one, where is this thing? How many colonies do we have on our ranch? How many colonies do my neighbors have on their ranches? And then primarily, how do we plant this thing? So we wanted to figure out some of my early work. What I did my master's on was looking at the cost effectiveness of using seed instead of container stock, just to see if it made more sense from a financial standpoint to just put the seeds in the ground instead of putting the seeds in a pot. And that had great results, and did my thesis on that, and have sort of just always been looking to that as a question. And with this species, it actually turned out to be a really important question. Okay, so we talked about this. So just more about the plant itself. Where does it grow? It really grows up out of the valley. So in our sort of first set of foothills, so if you sort of look at the map of California, you've got the Central Valley where Sacramento is, and then as you go east, you get this gentle rolling hills, and then you get up into the high mountains of the Sierra Nevada. So this plant goes from sort of that first set of foothills, which is what our ranch is, and up to, according to this, according to Cal Flora, 7,500 feet. On our ranch, it's in the shade, in the open, in rocky areas, in not rocky areas, on slopes, on flats. It does like to have its feet dry, which is odd for, being in a super dry environment. You would think that it would want some feet wet, but those other two milkweeds have already sort of occupied that space. So this guy likes it high and dry. So our two questions again, seed versus container stock, and then when. Some of the questions that had been going out there about this species was, do we need to plant it in the spring and then let it harden off and senesce in the fall? Do we plant it in the fall? So all those kind of questions. So we did a pretty exhaustive search, ATV bound search of our property and neighboring properties, and found a few colonies that we could collect seed from. You'll see this little bag, which took us a long time to find this. This is called this is like a wedding gift bag. Like if you're giving like some thank you, like, or rice, throwing rice at the wedding, something, right? You hold it in this little plastic mesh bag, and it works perfectly. You put it over the milkweed seed, because this plant, once it shatters, it's gone. It's not like some of the other species that we have. Narrowleaf milkweed produces several sets of seeds. If you miss one, you just go back a couple weeks later, there's another set of seeds. No, they all shatter. Once they're gone, they're gone. So with a species that's spread out, we have 5 colonies on our ranch spread out across 1,000 acres. So it's time consuming. It's not something you can go check every couple days to see if they're ready. So when we find them in this early case, this picture shows sort of right before they're ready. This is when we like to bag them. We try to not take anything more than 50%. So we leave at least 50% to continue dispersing naturally. We planted all of our seed was planted in the fall. It needs a 3-month stratification, which means it needs 3 months to be wet and cold. So in a nursery setting, you do that by like putting in a bag of moist vermiculite in the refrigerator for 3 months. But in reality, it's just mimicking what's happening out in nature, which is that it's sitting in damp soil all throughout the winter. So why not just do that directly? And so that's what we do. So we did this seeding, and I did all these different plots across the ranch and little rocky outcrops and protected with shade and all these different areas. And we got great germination in 2 back-to-back years, 2 consecutive years. Great germination and no survivors. Which if you spent, a day, it was 107 degrees at the ranch on May 30th. So how things ever got established here, I don't know. But we are working with an altered landscape. There's annual grasses instead of native grasses. There's climate change. There's a changed fire regime. There's cattle. So everything about these quote unquote natural systems is changed. But where we watered it, the seed did great. The containers, so some of the problems with containers, they don't overwinter well. So we had, because we were doing these multiple implementations, we did a fall planting, we did a spring planting, we overwintered part of the stock to do the planting in the spring. And of 40 containers, we only got 6 to come out. So, and some of the people that have done sort of a deeper dive on this species are working on better potting mixes and other strategies to help with that. But that was our experience. And then of that initial planting, only 7 of even the fall container planting are still there, versus the 25 seeded replicates that we did. We have 17 individuals from that. So a huge difference there. So this is a picture of one of the problems with planting the seed, the species from seed. So this would have been seed stratified in August, starting in August, and then potted out in January, and then planted in May. So they're pretty young, but because of the overwintering problems, you kind of have to plant them young. And this is an issue that has been ameliorated by some of these other experts. This thing called Jiffy pots is this little sort of bag that goes into the bigger container and sort of confines the root area a little bit smaller while they can still go into the bigger rooting area. The other thing about this is that the stems snapped off extremely easily. So the transplanting was insanely difficult because the soil's falling apart, the stems are snapping off, and it sort of became quite a challenge to plant the containers versus the seed. You scratch up a little area, you put the seed in, tube it, and you're good to go. So we did this in a paired side-by-side planting along irrigation in a very controlled part of the ranch where we have water and time and we can put eyes on it frequently. we did this planting in the 23-24 implementation year. Of course, they were all eaten by grasshoppers. So we replanted it this fall. And as of last week, for the containers, we have 18 out of 32. And for the seed, we have 15 out of 32. So really not much of a difference. Certainly the vigor, the container plants are definitely more vigorous. But from a standpoint of just pure survival, there's really no difference there. All right, so what the key takeaways in summary, here's a little look at those grasshoppers on it. Just gives me chills just to look at it. So I bought all these pantyhose at Walmart. I bought them out of pantyhose a couple weeks in a row as this grasshopper onslaught was happening last year, and they did work. So if you find yourself in a similar situation, grasshoppers are good only to a point, because when the leaves touch the pantyhose, they eat right through the pantyhose, and then once there's a hole, they all go in, and it's an ugly scene. All right, so the key takeaways. This pollinator island project, there really wasn't, as you saw on that results table, I guess it wasn't up very long, but the numbers from grazed versus ungrazed were really, we didn't get enough statistics on this to make anything statistically significant. So we're just talking about averages and pure sort of solid base numbers. The base numbers in the plots between grazing and ungrazing, there was basically no difference. There was also no difference in the species diversity, which I guess isn't super surprising when you consider that if a project fails because of lack of rain or grasshoppers, it doesn't care whether it's being grazed or not. And so I guess the takeaway from that is that, you know, one, we got these common species back on the ranch. We have 3 species that were locally extinct back onto our property and spread out across the landscape, not just here and there. They're all over the 700 acres. Higher priority species might need more special care. Year effects, please consider year effects. Please, grantors, offer more 5-year grant agreements. We need the time for monitoring. We need the time to maintain these sites. And then, of course, the milkweed stuff that I just talked about. So I think Jenna's coming on and telling me I'm out of time. So with that, I will stop. Thank you very much.
Thank you, Alex, so much. We'll hand it over to Alex and Jennifer and allow them to share their screen now. I love the reactions button. Thank you so much, everyone, for participating in that.
All right. Are you seeing my screen, Jenna?
Yes, looks great.
Awesome. Glad to hear it. Okay, get my windows in order here. Okay, all good. Well, thanks, Alex, for that great presentation. I'm the other Alex, just to make things confusing. And so we'll be transitioning to talking more about some of the Monarch Joint Ventures remote sensing projects. So I will get us started here. All right. So Alex talked a lot about planting milkweed and some of the on-the-ground efforts, but how do we track to see how we're doing in some of these milkweed efforts in planting? How do we know what's out there on the landscape already, and how do we measure whether our milkweed's coming up in some of these areas where we've done active restoration? Well, there's a lot of methods out there to measure milkweed density and habitat characteristics. I've just thrown a few of them up on the slide here. We've got the Pollinator Habitat Scorecard, which is a project of the Rights of Way as Habitat Working Group out of the University of Illinois, Chicago. There's the Integrated Monarch Monitoring Program, which is housed within the Monarch joint venture, which looks at both at habitat and at monarchs, adults and larvae. And there's a number of other monitoring methods, such as the Monarch Roadside Habitat Evaluator Tool, Wildlife Habitat Evaluation Guide, and even just ad hoc observations of what's out there on the landscape. Is there milkweed present? So as part of our remote sensing program, we're adding another tool to this toolbox of potential ways to monitor for milkweed and other pollinator habitat. So I'll dive a little bit more into our remote sensing program. So here at the Monarch Joint Venture, we started our remote sensing program in 2019. And our real goal with this program is to make it accessible. So we've done a lot of work here at the Monarch Joint Venture, but we really want to make this accessible to conservation practitioners across the landscape. And so we focused on using standard cameras and red, green, blue, or RGB imagery. And those are the type of photographs that your phone can take, that any camera can take, these basic red, green, blue images. And so that's what we focused on. There are a lot of other sensors out there, like LiDAR and multispectral, that can add a lot more information, but they're often more expensive, and so sometimes those are less accessible. So we've started off with these red, green, blue imagery as our focus. We want our surveys to be accurate and repeatable. In science, being able to repeat surveys is important, especially for tracking changes over time. And we wanted to make it easy to summarize data and share it so that we can actually use this information in research projects and in restoration and applying it on the landscape. And expandable. So we've started off focusing on a few species, but we want to make this something that can grow and be applied to other species and other habitat characteristics as well. All right, so some more details. We are using a technology platform called Poly. We've developed this in collaboration with Simple Business Automation, which is our technology partner. And so this online platform houses our flight plans, any images that are taken, and the data associated with those. And it also houses the algorithms that do the heavy lifting, and I'll talk more about those in detail. And the other nice thing about this platform is that it produces nice visuals, spreadsheets, and heat maps, and so that makes it easy to view the data and analyze it. All right, so what sort of drones are we using? So we kind of have two main categories of drones that we use in our remote sensing program. We've got some smaller drones, which fly at 30 feet above ground level. And we've also got some larger drones, which fly at 120 feet. So within the small drone category, we've got the DJI Phantom, which we've been using since 2019. We've also got the Anafi Parrot. And then in the larger drone category, we've got the Sony Airpeak, which we've been using since last year. And then this year, we're adding a new drone to our fleet, the FreeFly Astro. We're actually getting trained in on it this Thursday, so I haven't taken it out in the field yet. But these are the drones that we have that we use to do this monitoring. And I'll just point out that these three of these drones here are NDAA compliant, and those are drones that are compliant with the National Defense Authorization Act, and so they can be used on federal lands. And so that's a requirement for a lot of the projects that we do with federal partners, is that they have to be drones that are approved to fly on some of these federal lands. All right, how's it work? How have we developed our remote sensing algorithms? So to start off, we pick a species of interest. What are we interested in looking at? And the first one that we selected was common milkweed. It's the most common in the Midwest, important for monarch butterflies, and it's pretty easy to see, especially from above, from a drone. It's pretty distinctive. And so this was our initial species of interest that we started off with. We then assess the feasibility. Is it something that you can see with a drone? Which it was. And then we went out and collected a bunch of imagery of common milkweed. Here's a photo of my colleague Josiah flying a drone out collecting some imagery of the landscape that contained common milkweed. And then we went through and manually looked through all of those images and drew boxes around all of the common milkweed plants. So there's a lot of people hours looking through images, aerial imagery, but we had to train the model on what is milkweed and what is not milkweed. And then we test the model with new data, so take new imagery, and these computer algorithms go through and find the common milkweed, in this case, within the images. So here's an aerial image. You can see all these red boxes are around common milkweed plants. And then we made corrections. So if the model was detecting other species that were not milkweed, we had to retrain it, tell it what was what. And then we compared the model with the model output with humans. So we had some colleagues go out there and do some on-the-ground verification to make sure that what we were seeing in the aerial imagery and what the algorithm was detecting actually lined up with what was on the ground. So you can see here somebody flagging out all the individual common milkweed plants within this plot. And then testing in various field settings. So applying it to the landscape and testing it out in new locations. So we followed this process for a few algorithms that we're using right now. So I mentioned the common milkweed model, which looks at common milkweed. We've also got a relatively new model that looks at habitat in general and classifies the landscape into different habitat categories, looking at grasses versus dry vegetation versus flowering plants. And we're working on adding new models to our program. So we're currently focusing on our first invasive species model, which will look for Canada thistle. And we've heard from a lot of our partners, including those along rights of way, who go out and fly drones as part of some of their normal operations and monitoring. And there's been a lot of interest in using drones to survey for invasive species and catch some of these problems before they become big issues on the landscape as well. So I'll dive a little bit more into our common milkweed algorithm here. So how well does it work? When developing this model, we had to balance the recall and precision. So do we want the model to detect every single milkweed plant that's present on the landscape, although then it may overestimate and detect plants that aren't milkweed? Or do we want it to be really precise and only identify plants that the model is really confident are common milkweed? And so we had to balance between these. And so our current targets were 80% recall. So the model should detect 80% of the milkweed that's actually present on the landscape and 93% precision. And that means that 93% of the plants that the algorithm says are common milkweed are actually correctly identified as common milkweed. And here's a little graph going into some more details here. And so this was using 37 validation plots where biologists marked every milkweed stem. That was that picture I showed earlier with the flags. And we then compared this to the algorithm, which is represented by the blue line here, which detected 84% of the stems that the field biologists did and showed a very strong correlation with the field data. So the model is doing a pretty good job here compared to what the humans saw out in the field. And here's just some imagery of what this looks like from above. So here's the common milkweed model. These red boxes are around common milkweed plants. Here's a close-up. And here's our new habitat model. And so it's grouped the landscape into these different categories of dry plants versus grasses versus broadleaf plants, and then a number of other categories. We've got a number of output metrics. I'll go through these quickly here so I can turn this over to Jen. So some of our outputs are flight plan area. For our milkweed model, we get the number of milkweed stems detected and estimates of milkweed density in stems per acre and in which is the main number that a lot of restoration groups and milkweed researchers, this is kind of the baseline number that people use to compare areas. They're looking at stems per acre. So that's one of the numbers that we get. And then for the habitat model, we get percent cover of these various categories. All right, I'm going to turn this over to Jen, who's going to talk more about validating the model performance.
Absolutely. Thanks, Alex. We wanted to ensure that this was performing as well as expected in real life. So we vetted 404 images. So largely the same crew that was drawing milkweed boxes around the milkweed to begin with a couple years ago. Looked at some of the outputs from the model. In those images, 400 some images, the model identified over 3,700 milkweed stems. Within those images, we found almost 300 false positives, so something called milkweed that actually was not, and almost 1,000 false negatives, so those are missed milkweeds. And that actually turns out to be on target or about on target with our 80% recall and 93% precision. So that is about on par to what we were expecting. Those were all from the upper Midwest. Go ahead, Alex. Then we started exploring how the model performed in various situations. So we wanted to see if the model was affected by the milkweed density. We did notice, you'll see here, that there were more false positives, so more missed milkweed when the density was really high. Go ahead. And that was still, though, performing well within those typical milkweed densities we would expect to find based on several studies we looked at and scorecards of milkweed stem densities of interest. So that's that lower left quadrant down there where it's performing very well. And go ahead, advance. We had a couple outliers pointed to with the pink arrows there. And those outliers, for the most part, were really thousands and thousands of stems per acre, at which point you're not really losing much in terms of understanding your habitat context if you miss a few milkweeds in that image when you already have 1,500 or 5,600 stems per acre. Go ahead. It's also important for us to understand how much of the area to survey. This is related to two important things, time and money. So capturing fewer images can take less time, and processing fewer images can save money. Next, yes. So for example, we might be able to capture fewer images like what's shown here, as opposed to taking a picture in every single grid cell across the whole site. So to answer this, we flew 29 of our sites in the upper Midwest with 100% cover, took pictures over the whole thing, and then subsampled between 5 and 90% of those images, site by site, took 1,000 repeated measures from those subsamples, and then repeated that 100 times for each site. So that's what you're seeing here on this graph is the output from those 100 iterations. So there's 100 blue or red lines on here that are jumping around the true mean, the true milkweed stem density on this particular site called Ruby Red Peninsula, when we have a 5% sample size. If the line is blue, it means it is capturing that true mean. If the line is red, it means it's failing to capture that mean. So at 5% cover in this particular site, about 12 or 15 out of that 100 replicates fail to capture the true mean. So it's not great. Go ahead. When we add, let's see, more sample areas, those Y values are starting to home in around the mean. Now we have just a range that's around 50 to 225, as opposed to that much wider range we saw previously. And we have more blue lines, so we're more likely to capture that true mean. Go ahead. And this continues to improve as the sample size increases. So here up around 65% cover, that subsampling started to capture the true mean on pretty much all 100 iterations at almost all sites. So we're looking at this kind of starting point. Somewhere between 25 and 65% cover seems to be a sweet spot of where we might be able to capture images and process them to get an accurate representation of what's on site. Let's talk about variability too. So in a way, this is the patchiness of the milkweed on your site. So if go ahead and advance this now. If I'm subsampling my site, am I more likely to miss some variation on a small site or a bigger site? And if the site is bigger, will it be more variable or more patchy? We did not see a trend here. So the bigger sites didn't necessarily have more patchiness to them. Now, what about milkweed density? How does the density of milkweed itself on the site impact the variability of the milkweed density? So basically, are very milkweedy sites more variable or more patchy? And we found that, yes, there was a higher standard error around that milkweed density on the sites with higher densities. But we had to think about what makes sense and what matters here. For example, if you think about way on the right side of this graph here, which is showing the milkweed density on the x-axis and our standard error or variability on the y-axis, if you're way on the far end of the spectrum with 2,000 stems per acre and your standard error is 50, that's a relatively low standard error. You're hovering right there around 2,000 stems per acre. But if you're on the left side of this graph and you have a milkweed density of 75 stems per acre and a standard error of 50, that's a lot more meaningful. So we took our next step to focus on relative standard error. So when looking at relative standard error, we found that the relationship reversed. Relative to their means, the sites that had low milkweed density, here on the left side of the graph, had a pretty high variation in the images that we captured. So the question became, where do we want to prioritize this accuracy? We know that it's varying based on the density. So we looked at a lot of these scorecards and categories and cutoffs that actually Alex pointed out at the beginning in terms of the manual metrics and scorecards that people use to monitor. And we found that there were a lot more cutoffs to these categories and field realistic levels that were on the lower end of the spectrum, especially below 2,000 stems per acre. And so it seemed important to reduce that relative standard error, or basically increase our accuracy when the milkweed density is low, so we can fit into the correct pollinator scorecard category, for example. Here, long story short, we've got all these relative standard errors graphed out, and we've color-coded them. So the red and the orange typically are down toward the bottom. Those are sites that had a lot higher milkweed stem densities. And you can see they're at the bottom because they have a lower relative standard error. We're a little more accurate on those sites. The green ones towards the top have low stem densities. There's a lot more variability going on there. Next. So we used our data to split these into two categories. Long story short, I'll jump to the next slide. And we modeled some predicted values based on some high and low density categories. Now, so we have these two lines here, a red line indicating the predicted relative standard error on a site with high milkweed stem density. So that's 60 or more stems per acre. And a green line on there that is the predictive relative standard error on low density sites. If we think back to where we were landing, let's see, with somewhere around a 25 to 65% cover was starting to look pretty accurate to us. We were starting to capture the true mean with at least a 25% image cover. Let's look at the high category. So this red line at 25%. At 25% cover, we've got somewhere around a relative standard error of 0.34. That doesn't mean a whole lot other than the fact that we can figure out how much to sample on a low density site. So if you, follow that black line, we'd need to sample about 60% of our site to have the same accuracy where milkweed densities are low. Next slide. Basically, could fewer images do the job on a high density milkweed site? The answer is yes. We might be able to optimize around 25% image covered on a high density site and maybe 60% image coverage on a low density site. Fun part is you don't know your density before you survey it. So a potential solution could be to, go ahead, Alex, capture images over 100% of your site, process 25% of it, see where you're falling. And if you have really low density, you would process an additional subset of your images to get a more accurate measurement. Now, Alex is going to point out a couple comparisons between the remote sensing and some of those field methods that we talked about earlier.
Yeah, so we wanted to compare how our remote sensing compares to some of these pre-existing field methods. And so I'll highlight a few of those here. So the first comparison that we did was between the Integrated Monarch Monitoring Program, which surveys, has a standardized site area and then subsamples within that. So we're looking within these small white rectangles within that total area. And so we compared that area and the results from the Integrated Monarch Monitoring Program, and then flew that same area with our drones and compared the results of remote sensing in the same area. And so here's a graph. We've got stems per acre from remote sensing on the x-axis and stems per acre, the density estimate for the Integrated Monarch Monitoring Program on the y-axis. And in general, we found that the Integrated Monarch Monitoring Program had higher, about 2 times higher stem densities than our remote sensing surveys. And some of that is expected. We know that if you're on the ground looking at each individual plant, you're going to find a lot more milkweed than you will from remote sensing. If you think of, say, a real small milkweed plant that's buried in grass, there's no way you're going to see that from aerial imagery. However, this graph was influenced by this one outlier up here, which had really high stem densities by both methods. And so if we remove that outlier and focus in on this area, we've got this graph here that's zoomed in, and we can see a similar trend here with higher milkweed densities from the IMMP surveys compared to remote sensing. Here is about 4 times higher. And so here we're not necessarily comparing the accuracy of these two methods. They're both valid ways to measure milkweed densities, but we're going to do some more diving in and investigating these comparisons some more to see what gets us the best metrics for understanding the densities of milkweed on the landscape. In addition, we did some comparisons with the roadside scorecard, which is commonly used in the Monarch CCAA monitoring. And so that's a much smaller area. That's just 150 by 10-foot plot. And so we did the scorecard, we did remote sensing on top, and then we compared the IMMP plot that was in that area, which was a much smaller subset since it was only looking at 10 meter 1 meter square meter area subplots in that area. And so we can see here that the scorecards, there weren't any significant differences between these groups, but generally the scorecards had slightly higher milkweed density estimates than the other methods. But if you look here, you can see that the plots that had high milkweed densities with the scorecards also had high densities with remote sensing, and similar for these low plots with not much milkweed showed up similarly by both methods. And the IMMP is kind of targeted to looking at a larger area, and so this may not be the best way to compare that method, but we couldn't think of a better way to do it since it's looking at such a small area with the scorecard. So I'll just wrap up here with some of our kind of vision going forward. The remote sensing in general increases efficiency, so we can survey larger sites in less time compared to some of these methods that are looking at a small area, really closely at a small area, so it may be more cost effective. It's relatively comparable to field methods. Those sites that had high milkweed densities generally showed up in all surveys as survey methods as having high milkweed densities compared to sites with not much milkweed. So we'll continue looking more at some of these different comparisons. Remote sensing provides maps and raw data downloads, which makes it really easy to summarize your data quickly, as opposed to having to enter a bunch of data physically. It can provide specialized and automated calculations. We can process old images with the new models as we continue to develop more invasive species models and potentially other milkweed species in the future. We can go back to these flights that we've done in the past and process those images through these new models. And really our goal here is to allow others to be able to implement these models and use them out in the field, and test them in new locations, and make this available to the conservation community. So next steps, this summer we're expanding our sample size to new geographies. We're using new drones. We're getting other people and conservation partners involved who are using other drone models as well. We'll continue refining our existing models, the milkweed model and the habitat model, testing our milkweed model against some common lookalikes like dogbane to make sure that the model isn't detecting those. We'll be offering a lot of in-person trainings and online trainings to make this more widely accessible, and developing new models. I mentioned our Canada thistle model that we're working on, but we're hoping to add more as well. So if you're interested in getting involved, this is the contact for the Poly GPS website. You're welcome to reach out to them or contact us. If you're interested in getting involved, we're definitely looking for more people to apply this and open to ideas and suggestions. So thank you. Here's a photo taken from a drone of some of our remote sensing team, and here's our monitoring email address as well.
Thank you so much, Alex, Jen, and Alex, for your expertise and stories. I wanted to be sure you had plenty of time. We're over a little bit. I'm just going to ask a couple of quick questions. If y'all look at the Q&A, Alex P was able to get in and answer a bunch of the questions that y'all wrote to him. So please take a look at those. Alex, I will bring you on if there's anything specific you wanted to mention with any of those questions on camera. But just a couple of quick questions for Alex and Jen. Alex, what landscape features were most problematic for your models? For example, did you test it? Did it work in complex topography?
That's a great question. We are exploring that right now. Both of them were, the habitat model and the common milkweed model were based on upper Midwest, which doesn't have a lot of topography. So we're feeding some new like southern US or south central US imagery into there. It's actually right now more the problematic areas are where we lacked images over what you might call non-target areas. We don't have a lot of pictures of trees. We don't have a lot of pictures of water because those are not spaces that we fly over a lot. And so those tend to be more problematic categories, as opposed to the topography, which we haven't encountered obvious issues with yet and haven't done any hard analyses with in terms of what the topography looks like.
Great. And then one more question for y'all, and I know you kind of answered this a little bit, but would this model be able to be replicated in other areas to share with MJV? You know, you're talking about sharing it and such. What does that look like?
Yes, absolutely. Both those models, as they exist, are available to use in the Poly platform. That does require user accounts, but our intent is to encourage people to use that. The more data that are in there, the better we're able to maybe refine models. Additionally, as Alex said, as new models are generated, those will be made available or can be made available for others to use.
Awesome. Alex Palmerlee, is there anything you wanted to add on the Q&A questions that you answered for us?
I don't think so. I mean, I think there was one question I wasn't sure if it was for me. It was asking about the species of milkweed that's in the recording on my presentation, but it was Fascicularis cordifolia and Areocarpa. But I wasn't sure if they were talking about my presentation. So otherwise, yeah, I think it's all in the Q&A.
Sounds good. Thank you so much for answering those. I want to thank you all for your time and for your expertise. We will be sending a follow-up email by this Friday with the recording and then some answers to questions and other resources. We would love for you to fill out the feedback form. So if you're still here, I see 111 folks still here in attendance. Please Alyssa's going to throw that form into the chat. Please click on that and please take some time to fill that out for us. That really helps us to make this webinar series even better. And then we want to invite you to our next webinar, which is Tuesday, July 22nd, the science of radio tagging monarchs. We will be joined by folks from all over. Sean Butcher from the science director at Cape May Point Science Center and the senior R&D scientist at Cellular Tracking Technologies. Ashley Fisher, conservation biologist from Xerces Society. Lee Brown, assistant professor in biology from James Madison University. And Helen St. John, master's student in biology from James Madison University and in our science team, as well as myself, is very excited for this presentation. So I hope everyone can join us. And thank you so much, everyone, for being here, and have a lovely day. Bye.